Today, we're going to talk about how CRISPR is revolutionizing genetic engineering. Hey everyone! Welcome back to CHIZHIM Science Tech, where science meets the future. Today, we're diving deep into one of the most groundbreaking innovations in modern biology, a discovery that's reshaping medicine, agriculture, and the very fabric of life itself. This is the story of CRISPR, and how it's revolutionizing genetic engineering, one gene at a time. 1. What is CRISPR? To understand CRISPR, we need to go back to the battlefield between bacteria and viruses. Bacteria, like all living things, face threats. One of the biggest is bacteriophages, viruses that infect and destroy them. But bacteria aren't defenseless. They've evolved a primitive immune system, and CRISPR is at the heart of it. When a virus invades a bacterium, the bacterium captures a small snippet of the virus's DNA and stores it in its own genome. This collection of viral DNA fragments is what we call CRISPR arrays, essentially a memory bank of past infections. Later, if that same virus invades again, the bacterium transcribes a copy of the stored viral sequence into RNA and loads it into a protein called Cas9. This RNA guides Cas9 to the invader's matching DNA. Cas9 then cuts the viral DNA, rendering the virus harmless. So in essence, CRISPR equals the file cabinet storing viral DNA mugshots. Cas9 equals the security guard that cuts the intruder's DNA when recognized. This natural defense mechanism gave scientists an idea: what if we reprogram that system to edit any DNA we want? 2. From bacterial defense to genetic editing. The game-changing leap came in 2012. Scientists Jennifer Doudna and Emmanuel Charpentier figured out how to take CRISPR Cas9 from bacteria and adapt it for genome editing in other organisms, including humans. Here's the brilliance behind it. Instead of letting CRISPR only target viruses, scientists can now custom design the RNA guide to match any gene sequence in any cell. Once CRISPR finds its target, Cas9 cuts the DNA at that exact spot. Now, cutting the DNA isn't the only goal. What happens after the cut is crucial. Cells naturally try to repair broken DNA. During this process, scientists can knock out a gene by allowing the cell to make errors while fixing it. Insert a new piece of DNA by giving the cell a template for what to add. In short, CRISPR gives us a way to search, cut, and rewrite the code of life, with unprecedented precision. 3. How CRISPR works. Let's break it down into components. The CRISPR Cas9 system consists of two main parts. 1. Guide RNA. gRNA. This is a short, engineered RNA molecule. It acts like a GPS, telling Cas9 exactly where to go on the DNA strand. 2. Cas9 protein. Think of Cas9 as a pair of programmable scissors. Once the guide RNA locks onto the matching DNA sequence, Cas9 makes a cut. How it works. The guide RNA is created to match a particular gene. Cas9 and the guide RNA are delivered into a cell. The guide RNA finds its matching DNA. Cas9 cuts the DNA at that location. The cell attempts to repair the break. During this repair, scientists can disable genes, fix mutations, or add entirely new sequences. Imagine editing a sentence in Microsoft Word. You find the word error, highlight it, and either delete it or replace it. That's exactly what CRISPR does, but with A, T, C, and G, the 4 letters of DNA. This ability to precisely target and manipulate genetic code is why CRISPR is considered one of the most revolutionary tools in biotechnology. 4. CRISPR in medicine. Imagine curing diseases not with pills, but by correcting their genetic root causes. CRISPR is opening that door. Correcting genetic diseases. Sickle cell disease, caused by a single mutation in the hemoglobin gene. CRISPR can correct this in stem cells, which are then reintroduced into the patient's body. Beta thalassemia, another blood disorder that CRISPR is helping to fix, with successful clinical trials already underway. Leber congenital amaurosis, a form of inherited blindness, CRISPR is being used directly inside the eye to correct the faulty gene. Fighting cancer. CRISPR is being used to engineer immune cells, especially T cells, to better recognize and kill cancer cells. This is a major leap in personalized cancer immunotherapy. Treating HIV. Some scientists are experimenting with CRISPR to cut HIV DNA out of infected human cells, potentially erasing the virus from the body. This is a paradigm shift. We're no longer just treating symptoms, we're addressing the blueprint of disease itself. 5. CRISPR in agriculture. The world's growing population demands more resilient, nutritious, and sustainable food sources. CRISPR is reshaping the future of agriculture. Applications: disease-resistant crops, tomatoes resistant to bacterial spots, wheat immune to fungal rusts. Climate-resilient plants, crops that survive heat waves, drought, and floods. Nutrient-enriched foods, rice fortified with vitamin A, iron-enhanced wheat. What makes this different from traditional GMOs? GMOs often introduce foreign DNA. CRISPR, on the other hand, tweaks the plant's own DNA, making it more natural and often exempt from strict GMO regulations. This technology could significantly reduce pesticide use, boost global food security, and make farming more sustainable. 6. CRISPR in animals. Beyond plants and humans, CRISPR is being used in animal genetics, with both medical and ecological implications. Key applications: disease models. Scientists use CRISPR to create lab animals, especially mice, with human-like conditions for testing treatments. Livestock improvement. Pigs that don't carry diseases like PIAS, cows that grow more muscle, chickens resistant to avian flu. Gene drive in insects. CRISPR-based gene drives are used to alter populations of mosquitoes so they can't transmit malaria or dengue. Controversial example: glow-in-the-dark pigs have been created by inserting jellyfish DNA to study cell activity, showing just how precise CRISPR editing has become. 7. The ethical questions. With such power comes significant ethical dilemmas. CRISPR doesn't just offer hope, it also raises difficult questions. Should we edit the DNA of human embryos? Where do we draw the line between therapy and enhancement? Could CRISPR lead to designer babies, where traits like intelligence, height, or beauty are chosen? Who decides what's ethical? Who gets access? Real case. In 2018, Chinese scientist He Jiankui edited the embryos of twin girls to make them resistant to HIV. The backlash was enormous, as he crossed an unwritten global line by altering the germline, edits that can be passed to future generations. This event triggered international calls for regulations, moratoriums, and deeper public engagement with genetic ethics. 8. New advances in CRISPR technology. CRISPR-Cas9 was only the beginning. Scientists are now expanding the gene editing toolbox to make it safer, faster, and more precise. New tools. Base editing, edit single letters in DNA without making a double-strand break. Ideal for correcting point mutations. Prime editing, described as a word processor for DNA. Can search, cut, and replace targeted sequences with minimal damage. CRISPR and CRISPRi. Instead of editing the genome, these turn genes on or off, a major advancement for controlling gene expression. These refinements are essential for treating complex diseases where precision is everything. 9. CRISPR and the future of humanity. We're not just rewriting genomes, we're redefining what it means to be human. Possible future applications. Prevent all inherited genetic diseases. Bioengineered organs for transplants. Enhance resistance to pandemics. Resurrect extinct species like the woolly mammoth. Terraform crops for space colonization. But we must ask, are we prepared to handle these changes ethically and globally? 10. Final thoughts and call to action. CRISPR has opened the door to a new biological era, one where we don't just observe evolution, we participate in it. This isn't just a tool for scientists. It's a conversation for all of us about what kind of future we want to create. Like this video if it blew your mind, and subscribe to CHIZHIM Science Tech YouTube channel for more deep dives into the most powerful technologies shaping our future.